Hello dear little champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to finish the chapter that is Biotechnology Principles and Processes, and with this we can assure 20 marks in NEET. Yes, we saw that in the previous year how many questions came? 5 questions came from this chapter, which is not a small number. Yeah, and today we are going to talk about the production of the final gene product and also something about bioreactors, the different types of bioreactor which is given in your NCERT textbook. Okay, so this particular video will be more inclined to NCERT. Okay, this is a very short topic, so we will finish it ASAP. Okay, so we have studied about PBR 322 in the previous one, PUC8, which is also a vector, and also we saw how to make the host competent. Okay, not competent, competent. Okay, so you have electroporation, you have heat shock treatment, you have this, you have that, so many things coming together. So if you want to know about this, you can watch the previous video, and if you want the PDF for the same, you can join our WhatsApp channel, my dear cuties. Okay, number of questions was 5, and I remember giving you a homework. Let's see who all got it correct. Chalo. So in gene gun method used to introduce alien DNA into host cells. Okay, so there is a method that is known as gene gun, where with lots of velocity certain microparticles are inserted. Okay, are inserted into the cell and also inside the nucleus. So these microparticles are made up of something, some metal, and they are coated with DNA. So once they get inside, they are basically passing down the DNA, and this is known as gene gun. This is known as gene gun, or you also call it biolistics. Remember? Now this is done in the case of plant cells. Now that is a very important question that we did in the previous session. And what is that thing that we are using? What is a microparticle made up of? Children, it is tungsten or gold. Please remember, it's a very easy question, came in NEET 2023. It was being repeated. So we can expect something like this coming in the coming years as well. Yes. Chalo. So today's topic. So the basic need for culturing, if you want to culture a medium, the basically host. Okay, the host is a bacteria. Host is what, children? Host is a bacteria that is having the recombinant plasmid. That is having the recombinant, as in it has a plastid which has our gene of interest, what we wanted. Okay, so it is a recombinant bacteria that we are having, and we want to grow it up because we want to increase the number of the amount of the product that we are looking forward for. You know, right? The gene of interest which is there being inserted into the plasmid will be producing a protein, and this protein is of our interest. We want this protein, and for that we need to, for making more and more proteins, we need more and more host cells, that is basically the bacteria. Now it can be cultured inside a setup like this, but the basic needs we need to know about that. So we definitely need air so that they can do aerobic respiration and they can further do their own processes. Okay, so air is a must needed thing. Second thing, there should be a system to agitate. Okay, to mix things. There should be a system to stir things, to mix things. Why, children? Because you only imagine the air, suppose it is being provided from this side, okay, and then there is a feeding pump also, okay, where I am giving the nutrients. Okay, there is one more pump through which I am giving the nutrients needed for things to grow. Now suppose the nutrients are all accumulated on this side. Also the air is coming towards this side more, okay. Now if it stays this side, then only the bacteria surviving this side would be able to make it out, obviously. But over here, if there is a system to stir them, obviously the nutrient which has been put over here would be mixing along with things. Yeah, the air also, it will also be mixing along with things, and it will be really, really great. So we definitely want all this thing. We definitely want all this thing. Okay, that's a basic thing. So a culture medium, okay, which is rich in nutrients, a source for air, my dear children, a source for mixing the nutrients, the air, everything with the host bacteria and things like that. Apart from that also, you need so many things. There might be so many things, but this is the basic thing that we need. Okay, now we are going ahead. I hope you got it. So the small scale, there is 2 types of production. For example, I want something in very large quantities. At this point of time, I would be looking for big machines like bioreactors, about which we would be studying. As of now, I am looking for a small scale production. I don't want too much of product. In that case, what can be done? Okay, the things that can be done is of 2 types. One is batch culture and the second one is continuous culture. What happens in batch culture, my dear children, is that you have a setup being there where you are providing air, where you are mixing things, okay, where you are mixing things, it's all good. There is a stirrer, okay. Then you are providing the nutrients also. Everything is been done. Only thing that it lacks is there is a fixed batch, children. There is a fixed batch, fixed amount of nutrients, okay. So initially, if you see the growth of the bacteria which is there or the host which is there, it will increase because you are providing everything. You are providing everything. So first you have the lag phase where it slowly grows with time, okay, slow growth is there. Then there is a log phase that comes where it grows very much, and now you only think there is air, there is stirrer, there is nutrients. So obviously this bacteria will keep on using this thing, and there will be what, children? There will be waste products as well. There will be waste products as well. Now if you keep on providing nutrients, they will still produce more and more waste. They will produce more and more waste. So can we have that? No, that will be an issue. That will hinder all the processes that is happening within it. So obviously what will happen after a point of time when you are just providing fixed nutrients, okay, when you are providing just fixed nutrients, air and this there, it will reach up a stage where there is no increase in thing, and this stage is known as plateau stage. There is no increase in anything, okay. What is it? It is known as plateau stage. It's just constant there. And at this point of time, if you're not removing the important thing, if you're not removing the products and everything there, what will happen, children? And if you just if you just keep it on like this only, in this particular condition only, what will happen? Slowly it will decline. Slowly it will decline. So we do not want that. Okay, we do not prefer that to happen. Now there is one more type of culture where the thing is quite different. Over here, okay, the difference is that I am still providing air. There is a thing to stir things up. Okay, there is an effluent discharge. Effluent as in whatever is made, it has to move out. So this, whatever is made, the extra things that is being made over here, you call it effluent. In the effluent comes your product, okay, the protein that you want, also the waste materials that are being produced, everything comes, okay. So there should be an effluent discharge tube, and here you have fresh source of nutrients. It is just coming again and again, and things are moving out also, okay. But over here, as I said, it's a batch. You make it once, you take it out, and that's done, and then next batch will be going on. This is a continuous culture where you're continuously providing fresh nutrient, fresh medium, everything. But over here you have fixed nutrients. So initially I put it, that's all. With this nutrients they have to survive, and after they have made the product, you take that out and that's done. Over here I'm continuously providing, and obviously I'll get continuous effluent also, and from that I can remove my important things and the other things which are not needed for me. This is known as continuous culture because I'm providing continuously. Now the name of the batch culture is not being mentioned in your NCERT, but they are definitely telling about the continuous culture, so you should be knowing about it, okay. So over here, let's see, if any protein encoding gene is expressed in a heterologous host, okay, that is if a gene of interest is being expressed, this particular gene of interest is being expressed in a host, heterologous host as in that does not have anything to do with our gene of interest, okay. So basically if this host is expressing or basically making a protein, okay, it is known as recombinant protein. It's not their protein, it's our protein, our gene of interest that we have put over there. So this is what a recombinant protein that is being made over here, okay. Now the cells harboring cloned gene of interest may be grown on a small scale in the laboratory, okay. The cultures may be used for extracting the desired protein and then purifying it by different separation techniques. So after you have taken out the protein, obviously you have to purify it from any other source of waste material, as I said. Now the cells can be also multiplied in a continuous culture system wherein the used medium is drained out from one side while the fresh medium is added from the other to maintain the cells in their physiologically most active log or exponential phase. So over here, what is happening, children? I am continuously providing things. So what happens? It started from lag phase, it went up to log, and it goes in the log phase only. There's an exponential growth. Okay, log, or what do you call this growth as? Exponential growth. Now, if this question comes that in the case of continuous culture, what type of growth do you see? It is exponential growth. It is log phase that you continuously see. Okay, because you're continuously providing the medium and you're removing all the unimportant stuffs there. Now, this type of culturing method produces a larger biomass, leading to higher yields of desired protein. Obviously, this is preferred one because it will be producing more amount of proteins, more amount of product that we want. I hope this is clear. Okay, this is small scale production. Now, you have to think big. Okay, for thinking big, we need bioreactors, my dear children. Bioreactors, in your book, they have mentioned two types of bioreactors. Okay, one is simple stirred tank bioreactor. Okay, what do you call it? Stirred tank bioreactor. And the other one is sparged stirred tank bioreactor. So, this is simple. Okay, this is simple stirred tank. Whereas over here you have sparged stirred tank. What is the difference between them? You have to understand. This is the picture that is given. Okay, the notable differences that we can see is that over here, if you see, there's a curved surface. In the case of simple, there is a curved surface, whereas here it is not. Now, curved surface obviously provides a easy way to move around things, easy mixing and everything. So, that is fine. That is one difference. Apart from that, if you see over here, we have a system for sterile air and things like that. Okay, where you have a system for sterile air, and over here there is another mechanism where they are giving out air bubbles. And this air bubbles, if you see, it's in contact with the stirrer also. So, basically air bubble is moving out, and air bubble, it will be obviously moving out to different areas. Here the chances are that the air won't be properly mixed even though it has a stirrer. But over here, if you see, it is produced in the form of air bubbles, and these bubbles can move out very easily toward different areas, and it is just below the stirrer again, and it will help in the movement and easy movement and also easy distribution of the oxygen. Okay, so that's a good thing about sparged stirred tank, and over here you have simple stirred tank. I don't know if it makes sense or not, but still. Okay, so over here they have mentioned the foam breaker, flat bedded impeller, culture broth. So, this is the culture broth. There is steam for sterilization, there is pH control, there is motor for stirrer to move around. Okay, and over here what do you have, children? Over here, increased surface area for oxygen transfer. So, you have an area, a real long area, okay, which would be increasing the surface area for transfer of things. Bubbles dramatically increases the oxygen transfer area. If you see the bubbles, obviously it can move around really easily, and it will help in efficient transfer of oxygen molecules also. Okay, now let's read. So, to produce in large quantities, the development of bioreactors where large volumes of culture can be processed was required. Large volumes, what do you mean by that? Basically 100 to 1000 liters. Now, what kind of questions come from this? What is that something that is used for large scale production of things? Okay, so you should be knowing it's bioreactor. So, these small little question comes which we never imagine also coming. Okay, so that is the thing. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted to specific products. Now, I can give you a question. What is that thing that helps in conversion of raw materials? Okay, the normal chuma raw materials. What is that thing that helps in the conversion of raw materials to specific products or individual enzymes, etc.? What do you call this? You call this as bioreactors. Okay, now it can be microbial or a plant or an animal cell or a human cell, whatever is it, but it is being used. Now, a bioreactor provides the optimal conditions for achieving the desired product by providing optimal growth conditions. For example, temperature should be taken care of. All bacteria has an optimum temperature, an optimum pH. Okay, the amount of salt in them, everything should be maintained properly. The nutrients which are provided, that also has to be maintained properly. So, temperature, pH, substrate, salts and vitamins, all of these things have to be taken care of. Okay, so the growth conditions should be really good, then only things would work. Now, about stirred tank reactor, it is usually cylindrical or with a curved base. Okay, either cylindrical or with a curved base to facilitate the mixing of reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Okay, so stirrer, what is the use of stirrer, children? What is the use of stirrer? This is again a question that comes in NEET. It is to provide availability of oxygen It is to provide enough mixing and availability of oxygen. Now this is a question that usually get confused. People get confused. Is it for just mixing of nutrients and stuff like that? No, it is not just for mixing nutrients, but also for the availability of oxygen, and that is the most important task of a stirrer. This has come, and I am not joking. You get confused because everyone obviously could get confused with this particular thing, because that's not entirely correct, because you have so many uses of the stirrer, right? But still, the NEET people, they have asked, and that's why I am stressing on it again and again. Now alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system, an oxygen delivery system. Okay, it has an agitator system to move things, an oxygen delivery system, a foam control system, because obviously that could be very unhealthy if there is too much of foam being produced. It won't be a good survival strategy for the bacteria or any host. Now a temperature control system is there, a pH control system is there, and sampling ports, so that small volumes of the culture can be withdrawn periodically. So over here, what do we have? We have sampling ports as well. So little amount of things can be taken out and then seen whether it is being producing normally, or if we want it in little quantity, we can do for that also. Okay, so this is a general thing about bioreactors, and the questions which I said is the questions that I saw. Okay, nothing else, nothing new came there. Now if you are asked which is better, sparged one will be always better. Okay, because it will be spreading air in the form of air bubbles. Cool? Chalo. Now there's one more thing, that is downstream processing. So basically we know that there's a host which is producing our recombinant protein. Now the thing is, it is not very easy to get the recombinant protein alone, because you will be having, as I said, there will be waste materials. There will be waste materials. Okay, so basically the process of separation, okay, the process of separation of this thing, of the required protein. Okay, second thing, purification of this, fine. Also, after you have purified, you have to preserve it. Addition of preservatives all comes under downstream processing. All comes under downstream processing. So when you say that a particular recombinant protein is being separated or purified, or you're adding preservatives, you mean that it is undergoing what children downstream processing, and that should happen every time, because that's when you can obviously give it for drug making or whatever it is required for. After this, obviously there will be a quality check also. Obviously there will be a quality check, QC you call it, okay, whether it's good or not, can we use it or not, and everything. Fine. So these steps that you see over here, these three things comes under downstream processing. And now you cannot, when I show you the questions, you'll be like, my God, NEET can ask these easy type of questions as well. You will be in that zone. Okay, I'm pretty sure about that. Let's read it. So after completion of the biosynthetic stage, oh, I didn't tell you what do you mean by biosynthetic stage. So basically, biosynthetic stage means when the proteins are expressed, when the proteins, expression of proteins in the host. So when the proteins have been expressed itself in the host, what do you call this as? You call this as biosynthetic stage. So after that is being done, I am wanting to take out all these things out, separate things, purify my important things, and add preservatives to it. This is how things work. So after completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. Yeah. So the processes include separation and purification, which are collectively referred to as downstream processing. So separation and purification, I call this as downstream processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials, as in case of drugs. Strict quality control testing for each product is also required. The downstream processing and the quality control testing is required. That is what it means. Okay, that is what you mean by downstream processing. Now it's time for solving question. See through the questions, and with this we have completed the chapter. I want to tell you, tomorrow Saturday I'll be giving you one short video on the whole chapter. Okay, and we'll revise and we'll finish on Sunday. There's a mock test for this chapter, then class. Then we'll move on to the next chapter, which probably would be microbial or microbes and human welfare, probably that. Okay, that thing. Or it could be biotechnology also, I'll see. Okay, anyways. Now the first question. The process of separation and purification of express protein before marketing is called. Tell me children, tell me. NEET 2017 question. Can you imagine this easy question? Now if you're not aware about downstream processing, if you have just missed the part of the NCERT textbook, it becomes a huge loss. You see? And if you have read it, then you know the answer. This is the first question. Please tell me the answer. Second question. Stirred tank bioreactors have been designed for purification of product, addition of preservatives to help to the product, availability of oxygen throughout the process, ensuring anaerobic conditions in the culture vessel. What is the answer for this? Tell me. Again, something that I have pointed out and told that is important. Third, which of the following is not a component of downstream processing? Separation, purification, preservation, expression. Okay, expression as an expression of the proteins and stuff like that. Which of the following is not a component of downstream processing? NEET 2016 question. Fourth question. Which one of the following equipments is essentially required for growing microbes on a larger scale for industrial production of enzymes? So enzymes, please do not get confused, okay? Even if enzymes comes in the paper, enzymes are basically nothing but proteins, my dear children. So it's totally fine, okay? Now, so what is the answer? Bioreactor, BOD, incubator, sludge digester, industrial oven. What is the answer? This came in NEET 2019. Tell me the answer. Fifth, DNA precipitation out of a mixture of biomolecules can be achieved by the treatment with what? So this is something that is off the topic, but still I just thought of it and I just put it. So how can you precipitate the DNA out of a mixture? What is that something that will help you precipitate the DNA out of the mixture? Chilled chloroform, isopropanol, chilled ethanol, methanol at room temperature. I know I have discussed this question, but this is very important. So many questions have been coming around it and I don't want you to forget it. So tell me the answer. Okay, chalo. Let's do the questions. So the process of separation and purification of express protein before marketing is known as downstream processing, about which we just studied. Okay, stirred tank bioreactors have been designed for. Why do you need a stirring, children? You need the stirring because I told you it's availability of oxygen. You want to make the oxygen available throughout the container, throughout the reactor, which is why you have stirred tank bioreactors. And the best one is sparged stirred tank bioreactor. So this becomes the answer. Availability of oxygen throughout the proteins. I hope you are getting your answers correct. Now, which of the following is not a component of downstream processing? I told you. You want to separate, separate. You want to purify, purify. You want to preserve, preserve. This all comes under downstream processing. But expression, it is not something that comes under downstream processing. So this becomes the answer. Okay. Now, which one of the following equipments is essentially required for growing microbes on a larger scale for industrial production of enzymes? So bioreactor, BOD, incubator, sludge digester, industrial oven. Which is that something that you will need for growing microbes on a larger scale? Large scale, so obviously it has to be bioreactor. Okay. Now, DNA precipitation out of a mixture of biomolecules can be achieved by treatment with chilled ethanol. So whenever you put chilled ethanol, at that point of time, what happens, children? The DNA which is there, it will precipitate out. That's a good thing about chilled ethanol. Will you forget this? You should not be forgetting this. This is a very important thing. Okay, so please give a tick mark to it and I hope you got your answers correct. You see the year, it's not very far away. Okay, and questions have come this 2 years, sorry, in the same year, you see 2 questions coming around from this particular chapter. Well, there are a lot more. I didn't put them, but yes. Okay, so and I want to tell you, please give the mock test. It is a very important thing. There might be easy questions, it's okay. There might be easy questions, there might be medium difficulty questions, there might be very high difficulty questions, but you have to try it out. It will surely improve your confidence and it will surely give an idea of things. And there are a lot of times the questions has been repeated, my dear children. Now you cannot take that risk of not studying things which is very easy, right? That is a huge loss. So these things are very easy and I want you to concentrate in the class. I want you to try it out. This is not a very long lecture, right? This is just 25 minutes as of now. So yes. Now ready for homework, a quick homework. Chalo. Please try solving this question again. This is the same thing. This is the same thing. They have just twisted things out. I mean, like, they have just arranged things. So I hope you get this answer. So, stirred tank bioreactors have been designed for addition of preservatives to the product, purification of the product, ensuring anaerobic conditions in the culture vessel, availability of oxygen throughout the process. So, what is the thing? Stirred tank bioreactors have been designed for what, my dear children? Tell me in the comment section. And I hope that this particular thing was really useful for you. I hope that you have studied the whole chapter with me. If you still have doubts, please feel very free to put it down in the comment section. I will try to answer it. I might miss the live chat thing, but surely in the comment section, whatever good you say, whatever bad you say, I am reading it out. Okay, so please, if you have any doubt also, you can put. Okay, if you want to say something good, you can say. If you want to say something bad, that is also fine. Okay, but anyways, you have to interact with me. That matters a lot. Now, this chapter is very much important, and this chapter is the most easiest chapter that I have come across while teaching 12th as of now. I mean, easy and scoring. So that is quite a rare combination. In all the chapters, you have too much to learn, but over here, I didn't feel that there is too much to learn. So I think you can do really good in this chapter. So do not lose your 20 marks just because it's a new topic somewhere that is not entirely just like biology. It has some amount of technology also. So please do not ignore it and study it. Okay, so that's all. So subscribe to our channel for such good informations, for studying things, and for maintaining the preparation and everything. I hope you're having a good day. Please stay happy. Bye-bye. Take care.